Thank you for the very nice introduction, Louise, and thank you for inviting me here to talk to you today. It's a pleasure to be in front of such a diverse audience with so many different interests. So I'm going to be talking about mainly above ground, so crop health as a function of soil quality and soil management. And I'll be talking about leveraging novel technologies to increase the resilience of agricultural systems from molecular scales through to landscape scales. So we all know that the global demand for food is projected to increase and to approximately double by 2050. And on top of that, agricultural systems are facing increasing pressure from abiotic and biotic stresses. That means it's difficult to meet this target. Confounding that is the existing pressures on the availability of land means that we need to work with the land that we have available to us to sustainably enhance food production. So this is a schematic representing the cascading effects of climate change on food production. And it also integrates quite nicely the interactions between climate change, soil, and agriculture. So I'm going to focus here on the soil component. Firstly, it's quite a dense figure, but if you look at the top left-hand corner, we'll start with soil because that's the medium in which plants grow, and it's the way that many of their resource needs are met. So soil quality can be influenced by management, but it also can be affected by climate change. So we face problems related to soil erosion, disruption of microbial communities, and soil salinity, for example. And this means that we need to develop agricultural systems that are resilient to these kind of pressures and reduce the need for complex soil management techniques. So, in my opinion, we need to approach these kind of complex systems with a novel, multidisciplinary and multi-scale approach. And I'll start here with, I'll start here at the molecular level with a flavour of what's to come in the rest of the presentation and ask the question, is it possible to use genetic techniques to enhance the resilience of agricultural systems to stress? And this work is underpinned by the foundational work that was conducted by Professor Julie Gray at the University of Sheffield, who hypothesized that differences in stomatal characteristics would affect the ability of plants to regulate their water losses. So stomata are tiny microscopic pores in the epidermis of leaves that are surrounded by guard cells. And the movement of these guard cells affects the opening and closing of stomata and their ability to regulate those fluxes of water and also carbon dioxide between the leaves and the environment. And they're triggered by different environmental cues that you can see underneath these images, like low CO2, high light, for example. And Professor Julie Gray's team identified a gene, EPF1, that meant they could control the density of stomata on the leaves and restrict the stomatal development. So because we know that stomata regulate water, we wanted to know whether this kind of stomatal manipulation could be used within a climate change kind of framework and the stress framework to increase that kind of resilience to different stresses. And this work and the work in the next few slides was carried out by my brilliant postdoc, Dr. Bobby Kane at Sheffield. And he used these transgenic plants with the overexpression of EPF1 against a control of the rice variety IR64. And we can see that after this drought experiment, there was a cumulative water loss reduction in the transgenic plants by about 40%. And I'll come on to talk about why this might be in a couple of slides. But we do see that the stomatal characteristics, size, density, does affect water regulation. but it's difficult it's challenging in some countries or some circumstances to use genetic manipulation techniques, so transgenic plants. So Bobby wanted to know whether these kind of differences in stomatal characteristics were present in natural varieties that we could leverage. So he grew 72 different natural rice varieties and saw quite a large spread in these stomatal size density profiles, and selected 9 naturally occurring rice varieties with high to low stomatal density, and then the transgenic lines as well that are indicated in the green colour. So we can see there's a positive relationship between increasing stomatal density and GS max, which is stomatal conductance of water vapour, so the rate at which water is conducted from the stomata to the leaves. So soil salinity is a huge and increasing problem in many areas of the world, so we wanted to test to see whether these differences in stomatal conductance and water regulation could impact the resilience of these plants, these natural varieties, and also transgenic lines to be able to grow better in soil in saline conditions. So we used 5PS2 as an indicator of stress. So that's the photosynthetic efficiency of the plants. So the efficiency by which they can use the light that they absorb for photosynthesis. So essentially, the higher number, the less stressed they are. The lower number, the more stressed they are. So we applied 50 micromoles of saline solution to all 11 lines after 16 days after germination, and the speed at which they decline indicates the vulnerability to that saline conditions. And we can see that the green plants, so the lower stomatal density plants, the transgenic plants, are the most resistant, or amongst the most resistant to saline conditions, and the 5PS2 values stay highest, the longest, essentially. And we think that's because of the lower stomatal conductance. So essentially, there's less water that's being drawn up, so passing through the leaves, through the stomata, and there's less water being drawn up by mass flow from the soil. So there's a lower salt concentration in the leaves. Oops, sorry. We also wanted to test to see how these kind of differences in stomatal characteristics affected plants' response to temperature changes. So we conducted a temperature ramping experiment to see how quickly they responded. So we started off at ambient for rice temperatures, which was 30 degrees, and then we ramped up to 42 degrees. And we can see that the small size, high density plants, stomata plants, responded the quickest. So we can see drops in A, which is photosynthesis, quite rapidly after those temperatures started to increase. So the plants close their stomata to reduce water loss, and because the stomata are smaller, they can close much quicker. So that has implications for things like water use efficiency as well. So we've seen that these kind of molecular manipulations can affect how crops respond to stress, but this is all done in laboratory conditions using a lot of resources and manual measurements. And really we want to see how these kinds of manifestations can be tested in at field scales, in situ, and how this can be translated. And the way we do this is using remote sensing techniques. So there's a nice kind of schematic of how we move across scales here. So we've got the microscope, molecular level here, and then we'll move on to experiments where I talk about identifying the most appropriate remote sensing techniques for monitoring these kinds of changes that are physiological changes that are underpinned by stress. So we'll move on to some growth chamber experiments now. So this is under a different type of stress. So this is a nutrient stress now, and this is wheat plants. So we have a multifactorial experiment. So we look at competing nitrogen and or water stresses, because it doesn't really make sense to look at stress in isolation necessarily. We want to see how different types of interacting stress also affect plants, right? So we grew 128 plants, and at day 35, we split them into 2 sets. So the green plants were fertilised, and then the yellow plants were not fertilised. And then at day 60, they were split into a further 2 sets each, with 64 in each set, and then were droughted or not. And we can see here on the panels, the figures that show those kind of changes over time according to different measurement techniques. So we've got leaf area index, so the amount of leaf area per unit ground area, plant height, and then MTCI, which represents, it's a reflectance of vegetation index, but it represents chlorophyll content, it's a proxy for chlorophyll content. And then gas exchange, so pyrometry-based stomatoconductance measurements. And you can see how these different kind of sources of information are manifested through these different measurement techniques, right? So for the differences in fertilization, most measurement techniques kind of pick out those differences, right? So they manifest in the leaf area index, they manifest in the chlorophyll content, and also the pyrometry. But when we see the drought imposed, it's really only the gas exchange that shows how the plants are responding at that kind of instantaneous level. And actually, when we look at MTCI, which is the chlorophyll content, that doesn't start to change, or at least it doesn't show us any change until quite late on in the experiment. So the point here really is to guide your measurements. If you want to have an early detection, for example, is to take your measurements based on what is changing the most quickly, I suppose, and what's providing the most information. So this is an end of experiment kind of summation of the differences in chlorophyll content, nitrogen content, relative water content, and delta C13, which is an indicator of water use efficiency. And really the thing that I want to highlight on this slide is that it's the fertilized drought plants that show the poorest health, the poorest performance, and also the lowest yields, although the yield data isn't shown here. So when we look at the unfertilized plants, so the yellow boxes, actually whether they're subsequently droughted or not doesn't really matter, because the water demands on those plants are so low anyway. There's not enough nitrogen content to make those photosynthetic enzymes to drive photosynthesis to need the water. So it doesn't really matter if they're droughted. But when you start plants off with meeting their nitrogen needs and they're channeling mass flow through the soil and then they're droughted, then actually you see a much larger impact on the yields and on the health of the plant. So that's all very well and good as a kind of end of experiment, like this is how treatments manifest into final end of season products and whatnot. But actually, if we want to intervene, and if we want to have any kind of dynamic management intervention, then we need to model these kind of fluxes and responses to stress in real time. And the way we did this was through thermal remote sensing. So we use a modified surface energy balance approach, and really the two important parts of the equation are the temperature of the canopy and the temperature of there's a dry reference surface. Sometimes people say T air, so I've left that in the equation by accident. So T air or the dry reference surface is basically it's basically accounting for the amount of energy that's hitting the leaves, so it's kind of a normalizing factor. So it's the delta between leaf temperature or canopy temperature and the air temperature that matters. And this is because leaf temperature is inversely related to transpiration. So the cooler the leaves, the more the plants are transpiring because they evaporatively, the water loss evaporatively cools the leaves. Okay, so we've got, again, the kind of time series here of the different treatments over time and with different treatments. And on the left-hand side is, the right-hand side, is the pyrometry measurement. So that's leaf level, ground, if you like, validation measurements of what the actual fluxes of water are. The panel in the middle is our modelled estimates using these thermal imagery, using this thermal imagery. So it shows, it captures those kind of temporal dynamics quite nicely. And then we have the validation of the regression as well. So we have a fairly strong R-squared of 0.6. And more importantly than that, it's pretty much along the one-to-one line, and it doesn't show any kind of like deviations or bias related to the differences in leaf nitrogen content. So that's all very well and good. So we've done that in the controlled environment where we can control everything, the water, the climate, and whatnot. But the real challenge is how and we've identified the remote sensing techniques that we think are most suitable to answer different questions. But the real challenge is applying this in situ. So if we want to use this for phenotyping, or if we want to use this for precision agriculture, then we have to extend this to the field, right? So we use drones, and we worked on a variable treatment, nutrient treatment experiment in the UK. So there's 4 different treatments. There's a zero nitrogen, none, standard nitrogen. There's a little and often nitrogen, which is the standard nitrogen that's just split, essentially because we wanted to test whether we could reduce nitrogen runoff by giving the plants little amounts more often that they could actually use. And then a standard nitrogen with sulfur. And there's also, on top of that, a sugar and non-sugar treatment. So we flew drones over the field, and this is a mosaic of about 700 different drone images that are all stitched together. And just visually, we can see straight away the differences in the 0N field plots. And on the panels on the other side, there are measured ground level measurements of leaf area index, leaf chlorophyll content, photosynthesis, and VC max, which is photosynthetic capacity. And in nearly all cases, the zero nitrogen fertilizer showed significant differences to the other treatment. Generally, the regular N and sulfur treatment was presented the highest values of photosynthesis and the general highest values of plant health. And this is all very well good, and I mean, it's not too unexpected, right, that no fertilizer is going to reduce photosynthesis. But these are all taken using handheld measurements that are labor-intensive on a few leaves that are representative over the field, right? But if we want to use this in this kind of like dynamic precision agriculture kind of context, then we need to be able to create maps of these traits to guide applications of fertilizer, or in this case, fertilizer. And the way we do this is using optical remote sensing. So we use hyperspectral remote sensing. And these are characteristic vegetation reflectance spectra of the 4 treatments on the top panel, where the orange is zero nitrogen and the purple is the nitrogen sulfur application. And then the bottom panel is a relative reflectance. So we ratioed the 3 kind of less healthy vegetation treatments against the nitrogen sulfur, which was the healthiest. And we did this to kind of find out which bands were most suitable for pulling apart those different treatments. And we can see that they are in the visible wavelengths, so just around here. So 400 to 700 nanometers, so that's blue to red wavelengths. And these are wavelengths that we can resolve with our eyes. Anything beyond this, then we are not able to see. So we also see differences in 1400 and 1900, and these are water absorption bands. So we can diagnostically measure the presence of biochemical properties in the leaves based on the location and wavelength of their absorption feature. And by measuring the amount of light that they absorb, or the absorption feature depth, we can quantify how much of that property is within the leaf. Oops. And this highlights a couple of the known absorption features for these different molecules that we can use to leverage this information. So chlorophyll, protein, cellulose, lignin, and water bands. So from the drone imagery, we used a vegetation index. So we selected 3 bands that are known to be sensitive to chlorophyll content. And we used a common vegetation index, Merus terrestrial chlorophyll index, which is MTCI, and shows very strong relationships against measured leaf chlorophyll content. And then we use this statistical model to map chlorophyll content across the field. So the different treatments are highlighted by the boxes, and importantly, we can see really clear differences between those treatments, but we can also see differences within the treatments. Right, so this map is at about less than 5 centimetre in spatial resolution. So it gives us the opportunity to pinpoint almost individual plants or areas of the field if we wanted to guide where the farmers might need a fertiliser. So we also mapped, we used thermal drones as well to create a thermal mosaic in exactly the same way. And again, we can pick out those 0 nitrogen plots where the canopy temperature is higher because the transpiration is so much less. And when we compare the leaf temperature with measured stomatoconductance measurements from pyrometry measurements on the ground, we can see a remarkably high R², really, which is 0.6. So there's a strong relationship and there's a strong capability to use drones to provide different types of information about the plant physiological health. So chlorophyll takes a bit longer to respond, so it might take days to weeks to respond to drought or to nitrogen application, but the water fluxes is in real time, so you can start to understand quite quickly when plants might be stressed. So just propagating right through to yield now, we can see clear differences in grain size and overall yield. And this is quite a lot, but I just want to show the relationships. So these are correlations between traits that we measured and thermal and optical remote sensing products. And if we look at yield at the bottom and move across, we see that the relationship between yield and MTCI from the drone imagery is 0.97. So it's like incredibly high correlation between yield and chlorophyll content from drones. So it gives us that kind of like predictive element. So not only is it useful if we want to intervene, but it's also useful for yield prediction as well. So if we want to mitigate against or forecast yield losses from a kind of supply and demand point of view, nationally, internationally, this is super important. And that leads me nicely on to the final part of my talk, which is showing the potential of not only being able to do this at controlled environment levels in growth chambers at drones, but also from satellite applications. So we can leverage the power of satellite technology to do this kind of work over national to global scales. So this is just quickly some work that I did in Ontario. And it's on corn fields, and there's a 0 nitrogen application exclusion zone again, which is indicated by the box up here, and then the kind of measurement ground locations are here. And we took measurements every 10 days throughout June and September, 13 different sites, and we measured chlorophyll content, leaf area index, and reflectance on the ground. Then we paired this with Landsat 8 satellite data. So this is at 30 meters resolution. So this is a whole new level of complexity as we move across scales, right? So it's a trade-off. If we want to move to larger spatial extents, then we have a trade-off in the spatial resolution at which we can sample at. So that introduces complexity in how we deal with the data as well. We have to use process-based models to get back down to leaf level, but I won't go into that. So this is just one day, so we can see the field outline in the white boundaries with the 0N exclusion zone in the black, and we can see these kind of within field variations again, right? So And then this is for 4 dates throughout the growing season, and we can see the chlorophyll content slowly declining over time as the corn starts to senesce. But as I said, the power of this to look at, even from satellite data within field and variation in plant health, is huge. And the validation against measured chlorophyll content is again really high. The R-square is 0.87. So just to sum up, really, I think the complexity of the problems that we're facing in terms of agricultural resilience to different stresses, so abiotic and biotic stresses, in terms of soil degradation, soil management, which is, confounded by climate change pressures as well, needs a kind of multi-scale and multidisciplinary approach. So we can do work at the molecular level and we can understand those kind of plant physiological processes, but we also need remote sensing as well to monitor in real time and in situ how plants are responding to stress. We need to use remote sensing to map directly soil health as well, soil quality. And then we can input that also into a modelling framework, so we can model how crops are going to respond. We can forecast yield losses, but also we can use it in a predictive framework, so we can forecast under future climate scenarios which crop species, which crop cultivars are the most suitable to be grown in the future climate in a given geographical location. So integrating all of these different technologies and techniques, I think, is a really valuable way of increasing our agricultural resilience to future climate pressures. And with that, so I'd just like to thank my postdocs, Bobby Kane and Mohammed Khan, and various support that we've had for the field trials in particular. Thank you.
Thank you. That was wonderful experiments and very interesting results. So I'm coming to it not as a soil chemist, so forgive me, but my understanding about water and solute is that you can have you have both bound and free water, and in food chemistry we call the free water available water for water activity. So in your monitoring, I imagine you're monitoring the free water in your monitoring probes. I was wondering, could you refine that to an actual water activity, like from a scale from 0 to 1, like we do in food science, which dictates, metabolic activity and a variety of chemical reactions?
So we don't actually measure soil water directly. We measure the kind of, the mass flow of water through the leaves and how much they're transpiring.
Okay. So that would be free water.
Yeah. But in terms of beyond that, no. I have no idea.
Okay, thank you.
Okay.
So, hello. That was really fun. Creative applications, but I keep wondering, shouldn't somebody, instead of the way you're approaching it, here's a given location and a given crop, how do I optimize the results? Instead of that, shouldn't somebody look at and say, you shouldn't grow water-thirsty crops in this area, you should grow it where there's a natural higher rainfall?
Yeah, absolutely, yeah. So that's kind of, I guess that's what I ended on, or tried to end on, in terms of using this information within crop models that we can parameterize with future climate scenarios, and we understand where the rainfall will change and where the temperature will change, and we use this kind of species level, cultivar level, physiological information to say that, we shouldn't be growing crops there, we should be growing, different cultivars or whatnot in these locations. So that is exactly the point that we want to move on to.
I think we've got another question over here. But it will involve behaviour change though, won't it? Because you'll be telling people to change what they've
Absolutely,
done
yeah.
for centuries, potentially.
Yeah,
And
absolutely.
that's where you need the psychologists.
Yeah, well, I mean, the farmers are the people, they're like the canaries, right? They are the people on the ground that are seeing this and are doing, you know, already adjusting what they do in response to. So it's really, we should be talking, there to be a conversation, a mutual conversation.
Absolutely.
I have a question about the measurement of the timing of the water. Certainly, at different times in the crop growth cycle has major differences in terms of the impact. Could you speak to that a little bit in terms of your research? Did you look at various timing for when the drought occurred?
In terms of the plant developmental stages?
Yes.
No.
You know, for example, if the soil is wet when the crop is
Yeah.
planted versus if it's dry and then later, or if the drought comes when at various stages like of wheat production?
Yeah, no, that's a great question. We thought about it a lot, but we didn't really include developmental stage as a variable. We wanted to standardise for it, to be honest. There's only it's a logistical thing, I guess. There's only a certain amount of space we have available, and we didn't really want to introduce kind of different drought timings into the mix. But absolutely, that's crucial. Yeah, I agree.
Okay, fantastic. Thank you very much, Holly.
Thank you so much.